Hi, this is Mary from OpenHelix with this week's Tip of the Week. Today we're going to be talking about iRefWeb, a very nice protein-protein interaction resource, but I won't be able to cover everything in this short movie, so please be sure to check out our blog at blog.openhelix.com for additional details about iRefWeb, links to the site, links to additional information, and references that are associated with this tool as well. So let's move over quickly to iRefWeb to get started. Now, when you arrive at the iRefWeb resource, you'll find that there's an About page that tells you a lot about this project and links you to additional information. What I'm going to do is quickly move over to the search to get started to just give you an overview of the kinds of things that are available for you at iRefWeb. So here I'll click the Search tab, and this will load up the interface that gives you the access to the basic search mechanisms that you might want to get started. And here you'll see that you have a basic query interface here. You have filters, so you could set your search to do very specific things. You could refine it quite seriously with a lot of organism, source database, sources, types of interactions, and types of experimental details and things like this. You could start browsing around right from the home page here. There are over 400,000 protein-protein interactions in this database right now. It starts at 1, and you can go all the way up to 400,000 just by browsing around if you want. You can look by species. You can see that you have access to different species information here. You can see that there are different numbers of proteins involved in these interactions, and you could choose to look at higher numbers of interactions or lower numbers of interactions. You can see that there's information from a variety of PubMed sources as well. Different numbers of annotations from different databases, and all kinds of details that you might want to choose as you're performing your search. Now I'm just going to do one sample search to get started. BRCA1 human is the term that I'm going to specify. Now I could choose to set this up as a search for BRCA1 and with a organism filter, of course, but I'll just do this because you can use specific protein identifiers like this if you want. So here you would have access to the records that contain BRCA1 human. And now we've refined this a lot. There's only just over 250 here, but now you get to see a little bit more, and you can see that BRCA1 is one of the interacting proteins in this case, and you can see that there are some cases where they're human, mostly here they're yellow, but you can see that there's a case here, here's another one with another organism. Let's click that one to see what we would find if the iRefWeb interface gives us these details here, and what we find out is that this is an interaction between BRCA1 and a viral protein. Now let's take a look at this in the interaction graph. You can learn more details about the kind of evidence that's available here. But let's click one of these just to see the interacting graph here. And what you'll see is that you'll have access to this tool that loads up an interface that will give you all of the proteins that are available in a graphical representation. Now, it's only going to let me load 40 at a time, so I have to sort of trim this down a little bit. So let me say I want the ones that are with disease nodes. I'll just click those right now. Apply those to graph, and then those are going to appear. So here's the protein that I was interested in, and here are the proteins that are interacted with. They're all red because they all have disease associations, which is what we expected. So you can start moving around this graph. Of course, you have access to, let me unselect this, and you can start interacting with this. You can add the next layer of nodes out as well. So I can fetch the first neighbors of all of these, and it's going to load another layer of interactions, one layer out from all of these. Let me apply again. I'll choose again disease-related ones, but you don't have to do that. I'll apply those, and what I'm going to get is additional layers of protein information, protein-protein interaction information available as this graph will continue to load. And here you can start interacting with this and perform all kinds of explorations around this graph with a number of different features. And I encourage you to check out our blog post for more details about that. I don't have time to cover that right now. What I would like to do is show you a very cool aspect of this, and is that you can examine what kind of data is available from these protein-protein interactions based on the PubMed records. And I'm going to load up one of these sample ones to show you that the data that's in iRefWeb comes from a variety of different sources, and the sources collect different amounts of information. So here's one PubMed paper and the kinds of information that was loaded about protein-protein interactions in different databases. And so this is a very cool way to evaluate the kind of data that's out there in a given paper, and you can understand what kind of curation has gone on to get you that protein-protein interaction data. And this is a really, really interesting feature, I think, of iRefWeb that would help you to learn more about the curation process and protein-protein interactions and how they're stored in databases. So be sure to come to iRefWeb to check that out. Thanks very much for your time.